Good morning everyone. We are from group 4 and today we're gonna present regarding the amphibolic roles of TCA cycle in metabolism. Tricarboxylic acid cycle, also well known as Krebs cycle and citric acid cycle. The TCA cycle is the central metabolic hub of the cell. It is the final common pathway for the oxidation of fuel molecules such as amino acids, fatty acids and carbohydrates. TCA cycle is also amphibolic, which means it undergoes both anabolic reactions and catabolic reactions. This particular diagram describes the TCA cycle. The first step in TCA cycle is the formation of citrate. The TCA cycle begins with the condensation of a 4-carbon unit oxaloacetate and a 2-carbon unit the acetyl group of acetyl coenzyme A. Oxaloacetate reacts with acetyl coenzyme A and water to yield citrate and coenzyme A. Next, citrate is isomerized into isocitrate to enable the 6-carbon unit to undergo oxidative decarboxylation. Later part of the cycle, isocitrate undergoes dehydrogenation catalyzed by isocitrate dehydrogenase to form initially oxalosuccinate, which remains enzyme bound and undergoes decarboxylation to form alpha ketaglutarate. Soon, the reaction is followed by a second oxidative decarboxylase reaction where the alpha ketaglutarate forms succinyl coenzyme A. Succinyl coenzyme A is an energy-rich thioester compound. The cleavage of the thioester bond on succinyl coenzyme A is coupled to the reaction of phosphorylation of purine nucleoside diphosphate, usually known as GDP. This forms succinate. In the later part of the cycle, succinate is dehydrogenated by FAD to form fumarate. Next, fumarase catalyzes the addition of water across the double bond of fumarate, yielding malate. Finally, malate is converted to oxaloacetate by malate dehydrogenase, and this reaction requires NAD+.
Catabolic reaction in TCA cycle. Catabolism is the breakdown of large molecules into small molecules. Then, catabolic processes converts fuels into cellular energy. Fuel, in the form of carbohydrate or fats, produces carbon dioxide, water, and energy. For example, glucose produces carbon dioxide, water, and energy. In this flow chart, we have energy yielding nutrients such as carbohydrates, fats, and protein. They undergo catabolism, which releases GTP, ATP, and ADPH, FADH2, and produces energy poor end products, which is H2O and CO2. In this diagram, we refer to the Krebs cycles. 3 NADH molecules is released in the process of dehydrogenation of isocitrate, dehydrogenation of alpha ketoglutarate, and dehydration of malate. Next, dehydrogenation of isocitrate and dehydrogenation of alpha ketoglutarate also releases carbon dioxide molecules. Dehydrogenation of succinyl CoA releases energy in the form of GTP. Finally, the oxidation of succinate produces FADH2, which in the end accumulate all the end product. They have 3 NADH, 2 carbon dioxide, 1 energy which in the form of GTP, and 1 FADH.
In next next I will proceed on anabolic role of TCA cycle, which is gluconeogenesis. Okay, what is anabolic process? Anabolic process is a process that generates complex molecules from simple ones by using energy. For example, energy plus small molecules will produce complex molecules. Okay, as we know that TCA occurs in mitochondria while gluconeogenesis occurs in cytosol. Now I will explain the intermediate reaction between these two cycles. There are two mechanisms to transfer the molecules from TCA, which is in mitochondria, to gluconeogenesis in cytosol. Okay, first, phosphoenol pyruvate carboxylase will catalyze the decarboxylation of oxaloacetate to phosphoenol pyruvate with GTP as the phosphate donor. GTP will become GDP. This process occurs in mitochondria matrix. Next mechanism, which is mylate. Mylate from mitochondria is transferred to cytosol. Mylate is converted to oxaloacetate by using mylate dehydrogenase. Oxaloacetate is then converted to phosphoenol pyruvate and proceed to gluconeogenesis.
So next is role in fatty acid synthesis. Acetyl-CoA formed from pyruvate by the action of pyruvate dehydrogenase is the major substrate for long-chain fatty acid synthesis. So pyruvate dehydrogenase is a mitochondrial enzyme, and the mitochondrial membrane is impermeable to acetyl-CoA. So acetyl-CoA is transported into the cytosol from the mitochondria. When they are in the cytosol, they are cleaved in a reaction catalyzed by ATP citrate lyase. Acetyl-CoA can also be used in synthesis of acetylcholine, cholesterol, steroids, ketone bodies, and etc. Citrate is only available for transport out of mitochondria when acontinase is saturated with its substrate, and citrate cannot be channeled directly from citrate synthase onto aconitase. This ensures citrate is used for fatty acid synthesis only when there is an adequate amount of to ensure continued activity of the cycle. Citrate, besides transporting acetyl-CoA, the precursor of fatty acid outside the mitochondria, it also acts as a positive modifier for acetyl-CoA carboxylase enzyme, and this promotes de novo fatty acid synthesis. Citrate also regulates the rate of glycolysis by being a negative modifier for PFK1 enzyme. Thus, it inhibits glycolysis.
Okay, well, the next part is the anabolism or synthesis of non-essential amino acids such as glutamic acid and aspartic acid. Glutamic acid is done by glutamic dehydrogenase (GLGH), which converts alpha-ketoglutarate into glutamic acid through the process known as transamination, and alpha amino acid that is used in the biosynthesis of this type of protein. And glutamic acid has a conjugate base known as glutamate, which is the precursor of gamma-aminobutyric acid, a type of neurotransmitter. This neurotransmitter is widely used in major excitatory information transmitting pathway in the vertebrate brain. Next is aspartic acid. Aspartate is a non-essential protein being produced from oxaloacetate by transamination. Aspartate, which is a type of conjugate base on aspartic acid, stimulates NMDA receptors. L-aspartic acid is one of the two main ingredients of the artificial sweetener aspartame, along with L-phenylalanine. Okay, that's all from me. Thank you.
So one of the role in Amphibolic role of TCA cycle and metabolism is role in synthesis of purine and pyrimide. So first of all, I would like to talk about formation of glutamine. So glutamine is synthesized from glutamate by glutamine synthesis, which utilize an ATP to form glutamyl phosphate. This intermediate is attacked by ammonia as a nucleophile, giving glutamine an inorganic phosphate. Glutamine is used in carbon metabolism of carbohydrate. Then, glutamine is further processed into purines. Purine function directly as neurotransmitter, acting upon purinogenic receptor. Adenosine activate adenosine receptor. Two notable purines are adenine and guanine. So the second, I will talk about formation of pyrimide. Pyrimidine is an aromatic heterocyclic organic compound similar to pyridine. Aspartate donate one nitrogen atom in the biosynthesis of inosine, the precursor to the pyrimide bases. Three nuclear bases found in nucleic acid are cytosine, thymine, and uracil, which are pyrimidine derivative. So that's all. Thank you.
Biosynthesis of heme. There are two major sites of heme biosynthesis. The first one is liver, which synthesizes heme proteins, and second is the erythrocyte producing cells. In liver, the rate of heme synthesis is high, which responds to alterations in the cellular heme pool caused by fluctuating demands for heme proteins. In contrast, in erythroid cells, the rate is constant and matched to rate of globin synthesis. Formation of delta aminolevulinic acid. This diagram shows the formation of delta aminolevulinic acid. One glycine and one succinyl-CoA will condense to form ALA, which catalyzed by ALA synthase. This reaction requires pyridoxal phosphate (PLP) as a coenzyme. This reaction also produces CoA and carbon dioxide. This reaction is rate limiting step in porphyrin biosynthesis.